Hello students. Now after the completion of the section A, now move on to the next section, that is the section B. In this particular section there are 15 questions and you have to attempt maximum 10 questions. So let's start with the first question which is given in the board. Now here it is written that how many types of primary trisomics are possible in Datura stramonium where the 2n is equal to 24. Now what is the meaning of trisomy? Let's assume this one is a cell and this particular cell with 2n equals to 6. That means this particular cell with 3 sets of chromosomes. This one is a first set, this one is a second set, and this one is a third set. Now what will happen? Just have a look. In the second diagram, what will happen? In the second diagram, you will see there is a addition of a single chromosome in one set among these 3. And as a result of that, here we will see there is a 3 chromosomes are situated together and this set of the chromosomes is known as a trisomes, and the condition which results in the formation of the trisomes, this is known as a trisomy. Fine. Now here the trisomy observed in only one set, so it is known as a single trisomy. Similarly, like that, the double trisomy is also possible. Just have a look. This is the first set. This one is a second set and this one is a third set. So the trisomy observed in 2 sets, so it is known as a double trisomy. Similarly, like that, for this particular condition, triple trisomy is also possible. Like this. 1, 2, 3. 1, 2, 3 and 1, 2, 3. So this one is a triple trisomy. The chromosome number will be here, that's 2n equals to, here 2n equals to 7, here 2n equals to 8, and here that is the 2n equals to 9. So the maximum number of the trisomy is possible for a particular cell that equals to, that's a haploid set of the chromosome. If there is a 3 sets, that it is possible that maximum 3 trisomies are possible in that particular condition. So in this particular question, that how many types of primary trisomy are possible in Datura stramonium, if it is a 2n equals to 14, that means the haploid constitution is the N is equal to 12. So the answer will be the option 4, that's the 12. Okay. Now next move to our next question. Now in this particular question it is written that the percentage of the adenine in DNA isolated from the human liver is observed to be 30.7%. Then what is the expected percent of the thymine, guanine and cytosine? Okay. So we know that according to the Chargaff rule, we know that A is equals to T. According to the Chargaff rule, we know that the A equals to T and the G equals to C. So in this particular question, already it is given that the adenine is 30.7. So we know that according to the Chargaff rule, A equals to T. So definitely if the adenine is 30.7, then definitely the thymine is also 30.7. So A equals to 30.7, T equals to also 30.7. Now the rest of the nitrogen bases are either G or C. We know that definitely a DNA molecule is made up of 4 types of nitrogen bases, A, T, G, C. So just rest of the base are either G or it may be the C. And according to the Chargaff rule, we know that the G and C also present in an equal amount. So the correct answer will be the option 4, where T is a 30.7, G and C both are the 19.3%. Okay, so the answer will be, that's the 37, the answer will be, that's the 4. Okay, moving to the next question. If a hybrid DNA molecule labeled with the N15 is allowed to replicate twice in a normal culture medium, then the percentage of the hybrid DNA molecule will be. Now this type of question is constructed from the idea of the Michelson and Stalk experiment. Now in this particular question it is given that the hybrid DNA molecule where one strand is N15. That means, let us assume this one is a hybrid DNA molecule where one strand is a N15, the yellow one, let us assume that is the N15 strand, and the another strand, that's the N14, let us assume this one is a N14 strand. Now this particular DNA is allowed to replicate twice in a normal culture medium. What does it mean? It means that the normal culture medium means here the medium contains the nitrogen, and this nitrogen is a normal nitrogen, that's the N14 nitrogen. Fine. So the normal culture means the nitrogen which is present in the normal culture, that's N14. So what will happen? At the time of replication, first the parental strand get separated. Fine, these are the parental strands. Now the new strand gets synthesized, and at the time of the synthesis it uptake the normal nitrogen, that means N14 nitrogen. So as a result of that, after the first generation, 2 DNA molecule is produced. One N14, N15, and another one N14, N14. In the next step, these two strands again get separated. Okay, so this one is after first generation. Now after second generation, the newly synthesized strand, definitely it is N14. So as a result of that, this is the first strand, that's N14, These two also N14, and only a single strand with the hybrid DNA, that's N14, N15. So 1/4. So then the percentage of the hybrid DNA will be, that's the 25%. Okay, clear? So next, this is a very nice question. And here it is given that a particular template of bases in a coding sequence of the DNA is AAA. So let assume this one is a coding sequence. So the nitrogen base which is present in the coding sequence, that's A. So we know that both the strands of the DNA are complementary with each other. That means, let us assume this one is a template strand, then definitely the nitrogen bases which is present in the template strand is T, and T. Now when this template strand at the time of transcription produce the mRNA, let us assume this one is a mRNA molecule. So definitely the nitrogen base which is present in the mRNA molecule is opposite to the template strand. That means A, and A. And we know that the tRNA molecule, that means the anticodon part of the tRNA molecule, binds with the codon. The codon is present in the mRNA molecule. This one is a mRNA. Now the nascent mRNA molecule which is synthesized, the nitrogen bases which is present in the mRNA molecule is AAA. We know that the codon and the anticodon are opposite to each other. So definitely the anticodon will be, that's opposite to A, that's U, and U. Okay, so the answer will be UUU. Okay. Move to our next question. Now, a meteor fell on Earth. The sample analysis confirmed that 100 different types of amino acid, 100 different types of amino acids and 12 different types of nitrogenous bases. Okay? Each codon is probably. Now, we know that there are 4 nitrogen bases are there, and these 4 nitrogen bases are involved in the formation of different types of codons. Let's assume that the genetic code is monolith, so we can able to calculate the nature of the genetic code with the formula that's 4 to the power n. Here 4 signifies the nitrogen base and the n signifies that's the nature of the genetic code. Okay, whether it is a monolith or doublet or it is a triplet, so on and so forth. So, in this particular question, it is given that there are 12 different types of nitrogen bases. Okay, so definitely the formula will be that is a 12 to the power n. Fine. Now, if 12 to the power 1, let's assume the genetic code is monolith. If the genetic code will be, if it is monolith, then it will be 12 to the power 1, that means 12. So, 12 different types of codons may possible, and we know that one codon equals to one amino acid. So, 12 different types of amino acids are possible if the genetic code is monolith. Here it is given, it is responsible for 100 different types of amino acids. Fine. So, definitely the genetic code is not at all monolith. If let assume the genetic code is doublet or duplet, so it is 12 to the power 2. So, if it is duplet, then that's the 12 square. We know that the 12 square, that's 144. Fine. So, 144 different types of codons may be possible, and we know that one codon equals to one amino acid. So, if the genetic code is doublet or the duplet, then 144 amino acid may form. So, in this given question, it is written that's 100 different types of amino acid, that's less than 144. It means definitely the genetic code is duplet in nature. Okay, very nice question, and this type of questions are frequently asked in the different types of competitive examination. Okay, move to our next question. Observe the sequence given below. This is a sequence. The T is there. That means it is a DNA sequence. Okay. Now, which of the following will not lead to the frame shift mutation? Frame shift mutation. Now, what is frame shift mutation? Let us assume A, G, G C C C T. Let's assume this one is a DNA, a different sequence. Fine. From this particular DNA, the mRNA molecule which is produced is A. So it is U, 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 C, 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 G, 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 and A. Fine. So what do you mean by the reading frame? Reading frame means one codon is considered as a one reading frame. Fine. So this is the first reading frame. This one is a second reading frame. This one is a third reading frame. Okay. Now, if we delete one nitrogen base from the reading frame, okay, or if we add a nitrogen base in this particular sequence, there is a change in the reading frame, and this change in the reading frame is known as frame shift mutation. How it is possible? Let's assume from this mRNA molecule or from this DNA molecule, let's assume this A get deleted. Now this, if the A get deleted, so definitely here the U get deleted. So definitely now the reading frame will be shifted. That's UUC instead of UUU, CCG instead of CCC, and GGA instead of GGG. So there is a shift in the reading frame because we delete one nitrogen base from the intermediate position of DNA. Fine. So here it is given that observe the sequence given below and which of the following not lead to the frame shift mutation. Okay. So the correct answer will be thus option 4. That means the deletion of 7th, 8th and 9th bases together. Why? Because here this is a first reading frame 1, this one is a second reading frame 2, this one is a third reading frame, fourth reading frame, and the fifth reading frame. Now as we delete the whole codon togetherly, the remaining nitrogen bases remain same. There is no shift. Definitely one amino acid will less in the protein molecule produced from this particular DNA. Definitely it is that. But there is no shift in the total reading frame if we delete 7th, 8th and 9th nitrogen bases, because we are deleting a complete codon, not a single nitrogen base. So the correct answer will be that the deletion of the 7th, 8th and 9th bases together. Okay. Now move to our next question. This is another very simple type of question, that identify the chemical mutagen. As I have already discussed, that the mutagens are the agent which causing the mutation. There are several types of mutagens are available. One is a physical mutagen and another one is a chemical mutagen. So here the X-ray is an example, gamma ray is an example of the physical mutagen. X-ray is also a physical mutagen. So the chemical mutagen is option 3. Okay. The next question, the single cell protein Okay, which of the following statement is correct for this single cell protein? Now it is derived from the unicellular microorganism only. No, it is a false statement. This is a false statement. Why? Not only the unicellular organism as well as the multicellular organism, both are involved in the production of the single cell protein. We know that the fungus can also be used as a single cell protein. It is obtained from fungi and bryophyte. No, the bryophyte is not at all used in the production of the single cell protein. So this question, this statement is also wrong. The third, contains toxic proteins. No, this is also the false statement. So the correct one is, it processed to remove the excess nucleic acid, heavy metals and toxin. If these are not removed, all these metals get accumulated inside the body and it may develop several types of abnormalities. So the option 4 is a correct sentence with respect to the single cell protein. Okay. So this is the next question. Which of the following is an important biofertilizer in the rice field? What is biofertilizer? The biofertilizer, we know that the biofertilizer is a fertilizer which is used in the crop field just to increase the productivity. That's the biofertilizer. Fine. All these are the biofertilizers. So in the crop field, the biofertilizer which is used, that's the blue-green algae. Blue-green algae, or you can say it is a cyanobacteria, that's the Anabaena. So the correct answer will be the Anabaena. Okay, question number 45. It is given that the mutualism and the proto-cooperation can be represented as dash, whereas predation and the parasitism can be represented as dash. Now, we know that in case of the mutualism and proto-cooperation, both the members which are involved in this type of association, both of them get benefited. Fine. So it will be, that's a positive interaction. Whereas in case of predation and parasitism, fine, one member get benefited and another member get harmed. That means this type of interaction is a plus-minus interaction. So the answer will be, that's the option 2, plus and plus-minus interaction. Move to the next question. Here it is given that arrange correctly the sequence of xerophytic succession. We know that the xerophytic succession starts from the arid condition, barren land, where no lives are possible. From that particular area, the vegetation will sequentially grow by the process of the xerophytic succession. So, the first pioneer colonizer in a barren land is a lichen, is one. Okay. Next, it is followed by some small bryophytes, that means mosses. Now the mosses get replaced by the annual grasses, which is followed by the perennial grasses, herbaceous member, followed by shrub, and this is followed by the last, that is the climax stage. So the correct answer will be, first one is a 5, second one is a 3. So 5, 3, 5, 3. Okay, the next one is annual stage, that's mean 2. Okay, 2. Followed by that is a perennial, that means 4. 4. Followed by shrub, followed by climax. So the answer will be option 4. Okay. So move to the next question. What is the direction of movement of sugar in phloem? Now we know that the food material, that is the sugar, is moving from one place to another place through phloem, and the process in which the food material, carbohydrate type of food material which is produced inside the mesophyll cell is moving from one place to another place, and that particular process is known as a phloem loading and the unloading process, or we can say it is a mass flow. Okay. Now definitely in phloem the movement is always bidirectional. If I am talking about the ascent of sap, the movement is unidirectional, but the food transport through phloem is a bidirectional process. Okay. Now this is the next question. In some plants, the thalamus contributes to fruit formation. Such fruit are termed as. We know that the fruit is developed from fertilized ovary. Fine. Now in many cases it is observed that not only the ovary, the fruit is actually developed from the other parts which is not at all the ovary. Okay. So this type of fruit is considered as a false fruit. So in case of the false fruit, we will see that's the thalamus. That's the thalamus. This one is a thalamus. Is converted into fruit, as it is found in case of apple. Let us assume this one is an apple. This is a fruit with seeds, and we are having actually this fleshy part, and this fleshy part is considered as, that is a fleshy modified thalamus. So the correct answer will be, that's a false fruit. So this is a very nice question. Here it is given that the cross between these two parents, and as a result of that the progenies which are formed showing this type of phenotypic ratio. So it is asked that if this particular character is a polygenic trait, then what is the value of X? Now the first parent is capital A, capital B, small b, capital C, small c, which is crossed with another capital A, capital B, small b, capital C, small c. Now we know that these two parents at the time of sexual reproduction, they produce different types of gametes, and we know that the formula through which we can calculate the types of gametes, that's the 2 to the power n, where the n is a number of the heterozygotic condition. So here there is 1, 2, 3 heterozygotic conditions, so it is 2 to the power 3, 8 different types of gametes are formed. Here also 1, 2, 3, 2 to the power 3, that 8 different types of gametes are formed. Okay, now the number of progeny are 8 into 8, that 64 different types of progenies are formed. Okay, now these 64 types of progenies are segregated in a ratio of 1:6:X:20:X:1. So the summation of, that's the summation of the whole ratio is 64. So from this we can calculate that the value of 2x, it is 30, whereas the value of x is equals to 15. So the correct answer will be the option 3, that's 15. Okay, move to our last question. This is a statement based question. Read the following statement. This is an exceptionally large group of plants occurring in a wide range of habitat. They provide us with food, fodder, fuel, medicine, and several commercially important products. The range in size from tiny, almost microscopic, to tall trees over 100 meters long. So definitely this is a most diverse group of plant, and definitely this is the angiosperms. Fine. So this is a last question where we will see that the correct answer is the angiosperm. So this is the end of the section B and the end of the first video, discussion of the first video. So all the best students, and just stay tuned and just watch us in our next video.